new CAR T cells in solid tumors. He has moved to Barcelona, where now she is involved in the development of new CAR T cells in solid tumors. And finally, we have the opportunity to have Alejandro Madrigal with us. Alejandro, in the area of immunology and hematology, does not need a single word, but for those who are not or are more novel in this area of knowledge, let me just say that he's a master of masters nowadays, one of the persons with the widest knowledge and experience in the field of immunology, and he's also going to let us know more about new concepts on CAR T cells and the new projects he's involved in. So with no more delay, Michael, welcome to this meeting. It's a pleasure having you and listening to you for the next minutes, please.
Yeah, thank you for the very kind introduction. I hope you can hear me well.
Yes, we do.
Super, excellent. So, Professor Perez-Simon, thank you for the kind introduction. I would very much like to be with you. It's a gray autumn day here in Germany, and I hear it's very much warmer, and there was an excellent lunch buffet, so I would have loved to enjoy that together with you. But it's also good to be here with you online and share with you some thoughts about targets and technologies for CAR T cells. I also want to give a special shout out to Estefania Garcia Guerrero, who trained in my lab during part of her PhD. It's a pleasure to reconnect and continue our collaborative work, and maybe we get some new and additional ideas of what we can do together in the space of CAR T cell research. So the agenda for today, I've brought a few short stories and vignettes from our current research program, a short leisurely walk through some CAR T basics, then a look at CAR T cell therapy in multiple myeloma, a look at what we can maybe learn and do with the microbiome and artificial intelligence in order to improve the performance of CAR T cells. And then I will finish with a look at a few European network projects that, of course, also your center and your network is invited to participate in and to contribute in order to move this field forward. So it's now a decade that we've been witnessing the clinical development of CAR T cells. And also now in Europe, there's more activity, particularly actually in Spain. And of course, our common motivation and ambition is to move this field forward in Europe and work together. There's now 5 approved CAR T cell products with CD19 CARs and BCMA CAR T cells that we're actually using at our center here in Würzburg. We're in the middle of Germany, close to Frankfurt, if you haven't visited. And we're interested in now also witnessing how the approved BCMA CAR T products will be used in routine practice and how also the reimbursement will work as currently negotiations are ongoing with the healthcare insurances and the health payers that are always excited when they hear about CAR T and the big price tag associated with it. In any case, a brief reminder of what these synthetic immune receptors look like, trying to emulate the T cell receptor with this synthetic antigen binding domain that used to be derived from murine or rabbit hybridomas, and right now a strong effort is on developing fully human binding domains in order to reduce immunogenicity. And currently a lot of interest is in assessing the quality of the signal that is imparted by this signaling module composed of CD3 zeta to mimic signal 1 and the co-stimulatory domain to mimic signal 2. And it's clear that this receptor, even though it works, still has a lot of potential to evolve as due to the high affinity binding that happens with antibody-derived scFvs. The signaling in the context of high antigen density is quite explosive and may drive T cells into exhaustion, whereas in the context of low antigen density, it's often not enough to actually induce a productive immune response. So there's still a lot of work that needs to be done. And when you look at the complexity of the T cell receptor complex and all the signaling domains that take part here, there's still a lot to do and learn with CAR designs. But that's not the focus of today's talk, where I would like to jump right into our common interest in CAR T cell therapy for multiple myeloma. We're at the luxury situation to have multiple target antigens at our disposal. You're aware that BCMA is the most advanced. We have an interest in SLAMF7 due to the quite favorable clinical profile safety profile of the elotuzumab antibody that has been used for many years now. Noting though that SLAMF7 is not without problems, it is expressed on T cells themselves, so there is fratricide occurring that affects the ability to manufacture the cell product, but still leads, as I will show you, to the potential of inducing a potent anti-myeloma response. We've recently also shown that SLAMF7 is quite uniformly expressed in extramedullary lesions, also in patients that had undergone prior therapy with the elotuzumab antibody. We've not observed antigen down modulation or antigen loss, as we've recently shown can occur with BCMA, suggesting that SLAMF7 has some conceptual advantages in regards to the expression on myeloma cells. And we'll see how that plays out as we're currently doing a clinical trial. This is the Caramba study. It's supported in the Horizon 2020 program. This project has been going on for more than 3 years now. We have accomplished our first in-human treatment. The cell product that we're using has evolved also over several years of polishing and sharpening in the lab. There's a tailored spacer design in order to promote immune synapse formation between T cells and myeloma cells. We have a defined composition of helper and killer cells in order to enable a consistent pharmacokinetic. There's a safety switch, EGFRT, originally developed in Mike Jensen's lab at City of Hope that we can use to track the T cells, potentially also to delete with an anti-EGFR antibody, even though this has not been rigorously tested in humans. There's a humanized targeting domain, Hudecek in order to prevent the development of immune responses against the construct. And there is, as a technical vignette, also a virus-free gene transfer using the sleeping beauty transposon system. So here's two pieces of data from the preclinical IND program. So this cell product is quite potent. It lyses primary myeloma cells rapidly. And if you treat immunodeficient mice inoculated with myeloma xenografts, you can essentially cure the disease and have a 100% response rate. So these are similar benchmarks as what we've seen with CD19 CAR T cells in lymphoma models preclinically. So we're now conducting the Caramba trial. We do have approval now in 4 EU member states, Germany, Spain, Italy, and France. 2 of the sites have recruited patients, and we're currently opening the additional sites. Here are the consortium members. Our site in Würzburg is the clinical trial sponsor and also the coordinator of this project. The GMP manufacturing happens at the German Red Cross Blood Service in Frankfurt, like an hour away from here. They have a large clean room facility. And I'll just point out that Myeloma Patients Europe, as a patient advocacy group, is a full consortium member, has contributed to the writing of the clinical trial protocol, and is now helping to continuously educate and inform patients about innovative immunotherapy, including CAR T cells. A short look at the clinical trial protocol. Patients must have received at least 2 prior lines of treatment, including an anti-CD38 antibody, an IMiD, and a proteasome inhibitor. Patients that have received elotuzumab, and also patients that have undergone prior allogeneic bone marrow transplantation are eligible after a respective safety interval. And here's the clinical trial flow. Important to note that this is a fresh, non-cryopreserved cell product that we're using. The vein-to-vein time is only 16 days. So this is 14 days of manufacturing and 1 day each for the logistic from the treatment site to the GMP manufacturer and back. Product is administered in the inpatient setting, and patients are followed up then in the outpatient setting. Endpoints are around safety and determining the maximum tolerated dose in the phase 1 part, and then efficacy endpoints are included. That's a quite comprehensive exploratory research program, including also parameters like the quality of life during treatment and also the use of hospital resources. I can share with you that we've now completed the dose level 1. So this is 1 × 10⁵ CAR T cells per kilogram body weight, still quite low as a dose. So far, the signal has been quite favorable, no DLTs, CRS up to grade 1. We can track the T cells in peripheral blood. This is also coincident with the regression of serologic myeloma markers. And we're now entering into the dose level 2 with 3 × 10⁵ CAR T cells per kilogram body weight, where we also then expect a stronger efficacy signal. So as an interim conclusion, we have shown that it's possible to translate an innovative cell product, also one targeting a new antigen, in the academic setting to a first human clinical trial. I will point out, though, that this is possible with the support from the European Union, because this is essentially the only funding scheme that is big enough in order to pay for all the costs associated with such a clinical trial, including the GMP manufacturing and the procurement of vector in clinical grade quality. So far, the clinical signal has been quite favorable in terms of safety and also first indications of efficacy. And as a technical note, this is the first CAR-T product in Europe that uses transposon-based gene transfer. The second short story that I want to share with you is around the microbiome. And you're certainly aware that there have been several trials reporting on the association between the composition of the gastrointestinal microbial consortium and the outcome after immune checkpoint blockade. So the question is, are there compositions in the bacterial consortium or specific bacterial strains and or their metabolites that we can use in order to modulate the performance of antigen-specific T cells, and that we can potentially use as a supportive therapy in order to improve efficacy while maintaining safety, either in the form of specific pre or postbiotics, antibiotic treatment in order to shape a consortium, or the administration of specific bacterial strains in order to dominate such a bacterial consortium. And this has been our research question that we addressed together with colleagues in Marburg. And Mike Liu is the postdoc that worked on this and is now part of my team. So the experiments that we did is that we obtained fecal extracts from mice that were either germ-free, so no GI consortium, or held under specific pathogen-free conditions, so they have a bacterial consortium in the GI tract, and then co-incubated antigen-specific T cells. In this experiment, it was actually OVA as a model antigen in these extracts, and then looked at the magnitude of the T cell response. And we for sure found differences in cytokine production, also in lytic activity, survival, and proliferation. And then looked at which metabolites in these fecal extracts were mediating this effect. And we came across short chain fatty acids. They've been implicated as bacterial and microbial metabolites for quite a while. Our focus has been on propionate and butyrate, for example. We found that a short chain fatty acid pentanoate actually was the strongest inducer or mediator of this effect. And therefore followed up on this and went on to a tumor model, pancreatic cancer, in a syngeneic mouse model. We generated T cells targeting the ROR1 tumor antigen that is frequently expressed in pancreatic cancer, and then treated these CAR T cells during the manufacturing process with pentanoate, and compared the tumor response to T cells that had not been treated with pentanoate, or just NOC T cells not carrying a CAR. And indeed, the tumor response was markedly enhanced after the pentanoate treatment. The mechanism of action being, again, increased effector functions with increased cytokine production, but also increased infiltration of the tumor and survival of the T cells once they were in the tumor. We looked in the human microbiome, those bacteria that are in the GI tract, which bacteria are the strongest producers of pentanoate, and then butyrate, which was the second short chain fatty acid in line, and found Megasphaera mazaliensis, a bacterium that had not been on anyone's radar so far, as being the most potent producer of this. Mechanisms of action include regulation of mTOR. This is included in the manuscript. I will just point out here that the short chain fatty acids have HDAC inhibitory properties, both class 1 and class 2. We looked for an overlap with pharmacologic compounds that are HDAC inhibitors and found that still pentanoate and butyrate have a distinct pattern of how they inhibit distinct classes of HDACs. So it's not completely simulated by pharmacologic drugs, suggesting that also the use of pentanoate and butyrate would be rational in order to obtain that effect. And this is the conclusion, interim conclusion slide. So the data support the notion that there is an effect between the gastrointestinal microbial consortium, specific bacterial strains, and the metabolites that they produce between tumor antigen-specific T cells, including the engineered T cells that we use. And the data suggests that it may be possible to either modify the bacterial consortium or administer specific strains or metabolites in order to augment the intertumor effect. And this is work that we're currently doing also on example of hematologic cancers and multiple myeloma in order to increase the effect of, for example, the SLEMF7 CAR T cells I showed you earlier. Just as a note, what are we doing with these ROR1 CAR T cells? You see here, there's multiple prevalent epithelial cancers where ROR1 is expressed, including breast cancer, lung adenocarcinoma, ovarian cancer. So there's a strong interest to bring such a cell product into the clinic. We're currently working on what is called the Proof of Concept Initiative. That's a German initiative between the Fraunhofer, the Helmholtz Society, and German university hospitals. And we're currently establishing the GMP manufacturing for this cell product at the Fraunhofer ITSI. This is the GMP facility that has produced Kymriah for Novartis, for example, for the European market, and are currently applying for funding for clinical trials in solid tumors. Artificial intelligence is a big topic, and ideally, what we're dreaming of, that we kind of feed data from our preclinical development pipeline, and then the computer tells us what is the best receptor and what should we do clinically. This is, of course, this is not going to happen, but we can maybe use AI in order to inform specific aspects in either the CAR development process, the CAR manufacturing, and potentially the clinical use. And I will show you one project that we're currently invested in that may help us to get closer to this. But I will point out already this project 8Path. It's also a new project, is quite new. We just started in January with the ambition to establish an automated robotic manufacturing platform for CAR T cells that takes into account patient-specific biomarkers and uses AI algorithms in order to make it happen. Quite ambitious. The vision is in a very simple narrative here that to us as physicians and scientists deeply invested in CAR T may seem very simplistic, but there's also engineers, of course, IT and AI people involved. So this is why we said, here's a short visionary story of what we want to accomplish. There's a young soccer player experiencing a bleed during a soccer match, even the ball has not hit her. And acute leukemia is diagnosed. So workup in the hospital comprises sending a blood sample to this 8Path system that extracts specific biomarkers regarding the phenotype, activation status of her T cells, also the patient's history, and feeds it into the AI algorithm that then designs a manufacturing process for her CAR T cell product that is specifically catered for these T cells' needs. Product is delivered and patient is cured. So it's a kind of a little fairy tale here. But I think what it exposes quite nicely is that we want to look for biomarkers in patient T cells. Also want to look for specific aspects in the patient's history that tell us how well will a manufacturing campaign work, and when is a cell product at its optimal point of fitness, and potentially move away from the fixed time schedule of saying, okay, we're going to manufacture a CAR T product for 14 days, but rather say, when is the optimal time point? And in some patients that may be after 8 days, and others that may be after 10 days, when they have reached their climax, and where essentially, if you culture them longer, you overculture them and already reduce their potency and relative fitness. That's the one key aspect in that project. We're currently at the point where we are working on this AI, artificial intelligence use case, where we are looking at what biomarkers could be of potential interest and how can we get enough of these data and biomarkers in order to develop and feed an AI algorithm. It's going to be tremendous. It's not going to be enough to run like 100 small emulations of a manufacturing campaign. An AI algorithm will need much more. And we're currently thinking about how can we multiplex these manufacturing campaigns such that we can have enough material to feed into such an algorithm. If ever we make it, we will establish such a system at our site here in Würzburg. It will take a while in order to accomplish this. The last initiative I want to introduce to you is the T2Evolve initiative. It's innovative medicines initiative multi-stakeholder project that I'm coordinating together with Hélène Negret from Servier in France. So these are public-private partnership projects that have a common mission that was actually provided by the European Union, and then there were applicant consortia that proposed a specific work program and were then selected to execute this. And of course, the common mission here is to bring innovations in CAR and TCR transgenic T-cell therapy to the clinic faster. And in such a multi-stakeholder consortium, it's about creating common ground that everyone can use and walk on. And what we said, what we can accomplish together is to actually establish standards in preclinical and clinical development to make sure that more cell products make it through this translation bottleneck, and that we can actually enter into a cycle of productive bench to bedside and back learning. So the work program is here. So we have a strong emphasis on patient and healthcare provider information and education, and are currently collecting information materials, are curating those, and are trying to put them on a central hub such that more patients can get the information that they need and that they are seeking with regards to CAR T-cell therapy faster. It's also about creating new models to assess safety and efficacy, ideally moving away from animal testing in ex vivo models like tumor on the chip or organotypic tissue slices, as our colleagues in France, for example, use them. And it's about establishing gold standards for the manufacturing, the release, and also the clinical monitoring, such that the field can collectively learn faster. And I think this is exemplified also here. The idea is, if we're currently running 4 clinical trials, and every trial is running a different scheme for assessing pharmacokinetic, for example, then it will be difficult to learn between the trials. And it's almost impossible to have inter-trial comparability. However, if there was a standard methodology and a standard in time points, for example, when pharmacokinetic is determined, it would be possible to learn between the trials and increase the learning curve collectively as a field. And this is what we're currently doing to establish standards and methodologies that we are then curating such that they're ready to use for investigators that are interested in CAR T-cell therapy, including, of course, academic investigators that want to translate CAR T products from their in-house development pipelines. These consortia are very inclusive. There's, of course, a fixed number of consortium partners coming from academia, biotech, major pharma, patient representatives, policymakers, regulators also. But again, they're very inclusive. And if you're interested to contribute to the work that we do, there's the stakeholder community where everyone can make a contribution, but also then has the ability to use the output from the project. With this, I'm already at the end of my short presentation. So I've shown you our work, an update on our work targeting SLAMF7 in multiple myeloma. Some insights into how we can potentially use the microbiome to augment CAR T-cell therapy with specific bacterial metabolites. Our efforts of diving into artificial intelligence and trying to extract improvements in CAR T-cell therapy, and the T2Evolve EU project as a European initiative that is a multi-stakeholder initiative where we are trying to reach as many as stakeholders as possible. And this is why we're grateful to have the opportunity here to present it to the Rantecar consortium. I want to thank all the patients that are embarking on our clinical trials, the funding agencies. I want to point out that we have a few positions open at our program as we are currently expanding. And I want to also alert you to the 4th European CAR T-cell meeting that I have the pleasure to chair next year in February. The abstract deadline is end of November. There's going to be 3 abstract awards, each 10,000 K, that especially young researchers can then use to explore new ideas related to CAR T in the lab. So please submit heavily, and we hope to see you all on site in Nice, southern France, or online. Thank you.
Thank you, Michael, for this excellent presentation, and congratulations for all the work you have presented and you have been developing these last few years. There are time for a few questions from the audience.
Hi, Michael.
Hello, I can hear you, cannot see you, but hello.
Hello. You are not missing much, though. I have a question related to the AI project. You were talking about biomarkers, like in a kind of abstract way. I was wondering whether those markers could be like proteomic or genomic markers, and if so, don't we need like a huge beforehand study to know which markers may be relevant?
Yeah, no, the point is right, and I formulated this in the abstract way because this is of working through this what we call the AI use case at the moment to determine what in which area do we have the highest likelihood to find these biomarkers. Would that be just, would that be comprehensive phenotypic testing, or would we need to go all the way to like doing transcriptomic analysis? So this is currently work in progress, and I agree that it's a formidable task. And I'm showing this also because currently there's a strong interest, right, in using AI. Maybe sometimes our thinking of this is too simplistic, where we have the idea. Also, my understanding, right, to where we think, okay, let's just put some data into a computer, and computer is going to tell us what's going on. But these algorithms need immense amounts of data. And generating this data is one of the tasks that we're currently working on. And I agree. So it's not going to be simple.
Alejandro.
Mike, fantastic work and all the things that you are doing in network and collaboration, which is very important as we are doing with our project Rantecar, that I will talk a little bit on it. But you presented the use of ROR1 rather than ROR2, which are more expert, and you had to really even patent that one. So do you have any particular preference why in this case you use ROR1?
Yeah, so I have, the RAW family. It's this RAW1 and RAW2. So it's two analogs, right? And they have an interesting distribution in particular cancers. There are some cancers where they are concomitantly overexpressed, but there's also certain cancers where either RAW1 or RAW2 are expressed. So the RAW1 was actually the first target antigen and the first CAR that I've ever made. This is now like 12, 13 years ago. So it's very close to my heart, this antigen, and I want to see it being used clinically eventually. And the RAW1 it was first, discovered in chronic lymphocytic leukemia, actually, when by Lou Stout and his group, when they compared gene expression signatures between malignant lymphoma and normal B cells. And it was later then discovered that this is in a variety of solid cancers. It's on neuroblastoma and pediatric cancers, sarcoma, also triple negative breast cancer, ovarian cancers. So we're currently pursuing this in gynecologic cancers. So triple negative breast cancer and ovarian cancer are two indications that we're pursuing. And we also have a recent interest in certain endocrine tumors with the endocrine people here on site. The RAW2 often is in breast cancer, for example. When we've looked at patients, we found that those patients that are RAW1 high are RAW2 low, and vice versa. So it's almost like a puzzle that comes together. So there's also patients with breast cancer that would benefit from RAW2. Other indications include glioblastoma and renal cancer, for example, where RAW2 could be pursued. And we have a very potent salt product. The CAR that we have is also cross-reactive to mouse RAW2. So when we do the xenograft tumor models, it's at the same time also a safety test, and it looks very good. So the mice don't have any toxicology. So I think it's also an interesting candidate for clinical translation.
Thank you very much. I will talk a little bit about that. So
please.
Hi, Michael. You have presented very interesting data regarding the interaction between CAR Ts and the microbiota. And in fact, it's kind of very tricky because the same metabolites, such as butyrate, have been described in the post-transplant setting to play a role in immunotolerance. In example, the butyrate does increase the expression of FOXP3, increasing regulatory T cells, and decreasing the risk of graft-versus-host disease. So on the one hand, it seems to induce an immunotolerance, but on the other hand, it seems to improve the efficacy of CAR T cells. How does it work?
So you're pointing it at the right thing. So we also Another thing that we also see is that with these substances also have an effect on the tumor cells, right? So they may, in some cases, up or down regulate the tumor antigen or immune checkpoint inhibitors. So what we've come down to is that we say we need to essentially test this in a context-dependent manner. So for a specific antigen and CAR construct, it needs to be evaluated. And you're right, these substances have an effect on the CAR T cells themselves. Luckily, we found that they're not diminishing CAR expression and function. That's good. They will have an effect on other endogenous immune cells, and that needs to be accounted for. And they will have an effect on the tumor cells. And this is why it's still complex, even when we've broken it down to a single metabolite. And this, maybe when we extrapolate this now, how complex the microbiome by itself is, it maybe gives us a feeling of how complex it will be to actually modify the microbiome and get a positive effect out of this in the maybe on CAR T cells. So essentially, we've dissected now or deconvoluted the GI microbiome with thousands of bacterial species and brought it down to a single bacterial metabolite. And still, it's complex. And we would imagine that when we go back to the microbiome, it would be even more complex. What do you think?
Absolutely, absolutely. We have a lot to learn yet. And another question regarding the CAR T cell use in multiple myeloma. Unfortunately, so far the curves of event-free survival does not show the plateau we have seen in acute lymphoblastic leukemia or in lymphoma. What is your what do you guess? What is the reason for that? Is the immune cells from the patients which are more deteriorated? Is it the target that you are right now choosing a different one? What's going to be is there any specific target who makes the difference?
Yeah, I think you pointed out correctly already that it's probably a mixed bag of things, right? So we need to work systematically through multiple variables, try to improve and gradually come to the point where we see a plateau or similar activity as in ALL, for example. But it's probably a bit of everything. So I mean, we know that the T cells from myeloma patients, they are somewhat functionally impaired, especially if patients have undergone through multiple previous treatments. There's probably also additional mechanisms of resistance in myeloma cells, including when they are at different anatomical sites, extramedullary, for example. And there's something to the target, mainly antigen down modulation, or even this homozygous loss, as we've recently described for BCMA, has not been described for CD19 in lymphoma. It has been observed, in acute lymphoblastic leukemia with the exon splicing, where the epitope for the CAR gets lost. So there's particularities that need to be learned in every disease. I'm quite optimistic that we can still improve with what we see with BCMA CAR T products in multiple myeloma if we go to earlier lines, if we use maybe combination therapies that force myeloma cells to sustain the BCMA expression, for example. But probably we also need to think hard about dual antigen targeting. I guess multiple myeloma would be an exemplary disease where this can be of benefit.
There is another question from the audience, Estefania.
Hello, Estefania.
Can you hear me now?
Yeah.
Excellent talk. Thank you, Michael. It's a pleasure to hear you. Going back to the platform artificial intelligence, are you also considering to search these biomarkers in the leukapheresis, not only in the T cells when you are manufacturing, also in the leukapheresis in order to have a good product, we could say, or in order to
You're pointing it out correctly. So it would be ideal. We're doing both. We're going to look in the leukapheresis product at the start in order to kind of being able to predict the future. Whether that will be possible, I don't know. And then also look at during the manufacturing to look at biomarkers and say, okay, when do we think the cell product really has reached its optimal time point? So we're doing both.
Okay, so we have to move forward. Michael, thank you very much for your presentation, and I look forward to meeting you physically here in Sevilla soon.
Yeah, please have it at some time when it's nice and warm, warmer than in Germany.
Avoid July and August.
Okay, I will stay with you for a few more minutes. At 5 p.m. I have to go to a faculty meeting here, but it was a pleasure to be with you virtually. Take good care of Estefania. She's wonderful, and I hope to expand the collaborative work. Right. So thank you.
Great. Thank you. Thank you very much.